Hello, everyone, and welcome to today's event. The Leukemia and Lymphoma Society of Canada is happy to present this webcast on Scanning the Horizon, AI and the Future of Blood Cancer Treatment. My name is Paul O'Connell. I'm the Research Program Director at the Leukemia and Lymphoma Society of Canada, and I will moderate this online event. So during this presentation, you will listen to 4 grant winners from last year's Blood Cancer and AI Catalyst Grant Competition speak about their work, which focuses on the use of artificial intelligence or machine learning and blood cancer. I will be introducing each of these speakers in a few minutes, but first a few housekeeping notes. We invite you to type your questions and comments into the Q&A box at the bottom center of the Zoom window throughout this presentation. An LLSC staff member will monitor the questions, and I will ask some of them to the speakers at the end of the presentation. Due to the large number of participants and limited time, we may not be able to get to all of your questions. Also note that there will be no personal health questions answered during this webcast. One further note is that if you have a question that you would like to direct to a particular speaker, please write the name of that speaker in at the beginning of your question so that we know know where to direct the question to. Today's presentation will be recorded, and therefore you can rewatch it again on our website. As we come together from across Canada, we recognize that we are on lands cared for by many Indigenous nations. We honor the First Nations, Inuit, and Métis people whose deep connections to these lands enrich our community. We value Indigenous voices and are working to ensure that they are truly represented in our leadership and staff. At the Leukemia and Lymphoma Society of Canada, our commitment to diversity, inclusion, belonging, and health equity guides how we work to support and uplift everyone affected by blood cancers. Before we present the first speaker, I want to share the important mission of our organizations. Are you looking for specific programs or resources but not sure where to find them? We are here to help. You can connect with a community services lead in your region to receive personalized assistance and get connected to resources in your area. Reach out by calling toll-free 1-833-222-4884. We invite you to visit our website where you will find tailored information and resources in multimedia formats to address the variety of needs and preferences. This includes fact sheets and booklets, podcasts, animated videos, and recordings of all of our webcasts. Visit bloodcancers.ca to read, listen, and watch all of these great resources. So before we turn it over to our expert speakers, I want to briefly introduce you to a powerful force that is reshaping the future of medicine, and that is artificial intelligence, and its potential to revolutionize the way we diagnose and treat blood cancers. Early diagnosis and prediction of blood cancers can significantly lower mortality rates and increase survival. As the amount of biomedical data grows exponentially, from genomic sequences to pathology images to electronic health records, researchers now have an unprecedented opportunity to apply computational tools to uncover new insights and deepen our understanding of these complex diseases. Among these tools, artificial intelligence and machine learning stand out as some of the most promising. So what exactly is artificial intelligence? And no, we're not talking about ChatGPT or Copilot or Gemini. At its core, AI, or artificial intelligence, or AI, refers to the ability of machines to mimic human intelligence, to learn from data, recognize patterns, solve problems, and even adapt over time. It's not just about automation, it's about enabling machines to make informed decisions. A key branch of AI is machine learning, where algorithms are trained on large data sets to make predictions or classifications without being explicitly programmed for every scenario. In healthcare, this means AI can analyze massive volumes of complex data faster and often more accurately than humans can. Now imagine applying this technology to one of the most complex and life-threatening diseases, that is blood cancers. This group of cancers, which includes leukemia, lymphoma, myeloma, myeloproliferative neoplasms, and myelodysplastic syndromes, affects the blood, bone marrow, and lymphatic system. Diagnosing these cancers is often difficult, requiring expert interpretation of blood tests, bone marrow biopsies, and genetic data. But the use of AI is changing that. Recent clinical studies have shown that AI-powered tools can detect blood cancers with higher accuracy and even higher specificity in certain types, like lymphoma. These systems can analyze thousands of variables from flow cytometry, genetic sequencing, and pathology, far more than a human could process at one time, potentially leading to earlier and more accurate diagnoses. AI is also transforming how we treat these diseases. Machine learning models can predict how a patient might respond to chemotherapy based on their genetic mutations and clinical history. This allows oncologists to potentially tailor therapies to the individual, reducing trial and error and improving outcomes. AI is also being used to identify new drug targets and repurpose existing medications, accelerating the pace of discovery. The Leukemia and Lymphoma Society of Canada is actively investing in this future through initiatives like the Blood Cancer and AI Catalyst Grant, which was offered last year. We are funding bold, forward-thinking research that aims to push the boundaries of what is possible. This one-year proof of concept grant was designed to explore the capabilities of AI and machine learning in advancing blood cancer research, diagnosis, and treatment. Rather than funding incremental progress, these grants are meant to push the needle, supporting projects that challenge conventional approaches and open new frontiers in prevention, early detection, and care. We hope to find new ways to prevent blood cancers, improve survival through early detection when the disease is most treatable, and develop more precise and effective treatments with fewer harmful side effects by combining scientific, clinical, and patient expertise that integrates the power of new technologies and innovation. So today, we have 4 blood cancer researchers that were funded through this competition that will now share how their innovative work, which is supported by LLSC, is exploring the role of AI in exciting applications across different types of blood cancers. In the fight against blood cancers, AI offers as a powerful ally, one that can help us diagnose earlier, treat smarter, and ultimately save more lives. So with that, I'm honored to turn things over to our esteemed guests. And I believe first we have is Dr. David Andrews. So Dr. David Andrews is a senior scientist in biological sciences at Sunnybrook Research Institute. He's a professor of biochemistry and medical biophysics at University of Toronto. He has been a Tier 1 Canada Research Chair in Membrane Biogenesis, a Novartis Scholar, a president of the Society for Biomolecular Imaging and Informatics, and a member of the editorial board of Cell Death and Differentiation. Dr. Andrews will talk about his research, which is entitled Primary Cells Identify CLL Patient Cohorts with Distinct Drug Responses. Over to you, Dr. Andrews.
Oh, your microphone you're on mute, Dr. Andrews.
Yep, got it.
Good.
Okay. So this first slide is sort of an overview of what it is that we're trying to do. And it comes from sort of a pragmatic view. We were involved in genomic sequencing efforts and looking at mutations. And either the results were that there were too many to interpret, or if we had something interpretable and a driver mutation, there was no drug for it. So the idea was, could we use patient cells and identify the optimum drug combination for an individual patient? The problem with that, and that's what's now often termed hyper-personalized medicine, is that you can say, okay, this is the best drug combination for you, and you give it to the patient, and you would never know whether or not it was right. You don't know if they would have done better on something else or worse. And so we decided that really what we needed was small cohorts, because with small cohorts, it would be possible to do clinical trials. So the idea was to find cohorts in a pragmatic way. So we take cells from patients, we do something called conditional reprogramming to enrich for the driver cells, and then we image them and identify patient cohorts and link that to drug responses. So I can have the next slide. So this is the conditional reprogramming part. And so what you do is you use cytokines and things. It turns out that this whole idea of a cancer stem cell it probably underestimates the pluripotency of, or potency of many cells. Many cells can act as driver cells and can drive a new tumor. They just need to be told to do so. So that's what we do, is instruct them to behave like driver cells as if they're in the bone marrow or other proliferation centers in the body. We then take those cells in the next slide. Next. They look like this. And so this is a bunch of cells from a CLL patient. We're doing this with both chronic lymphocytic leukemia and AML. I'm only going to talk about CLL because we're further along with that disease. So I have added those little shapes that are on this image. And inside those boxes, or circles, or pentagrams, or triangles, are things that I think look like each other and look different than the other cells. Sometimes they make little clusters. Sometimes you'll see from the dyes here, these are dyes that I invented, which don't change the transcriptome of the cells. They're completely non-toxic, and so they're very useful for imaging patient cells. So it's very heterogeneous. So what can we do with that? Well, we can push the cells one way or another way to get a physiological response that we can interpret. The next slide. So if we blow this up, you can see that some of the cells have, these yellow sort of tails sticking out of them. Other ones have formed a little cluster where the yellow bits are together. The yellow is metabolic activity. The red is stress levels, and there's other information in the red that the AI can pull out. And then the blue is the nucleus for each cell. Next one. So here what we've done is to add drugs that we know cause a specific phenotype. So we have ones that cause quiescence, ones that cause stress, ones that cause activation, things like that. And then we train the AI algorithms on all of these different physiological responses from the cells. And then we can ask in any patient, what kind of cells do they have? Next slide. Next. So here's what you see. So the DMSO is just the normal, not healthy because they're cancer cells, but the cells that are conditionally reprogrammed, so they believe they're in the bone marrow. And then the different colors are different kinds of cells. So the purple ones are activated, the yellow ones are stressed, and the two that are the most different from everything are the black ones and the green ones, and those are both dead. But they have died by different pathways. So we can even identify which pathway a cell used to die by looking at the images of the cells. The one I want to point out is the stressed one, because we're going to come back to that. And also it shows you some of the power of this kind of technique. We use a drug called tunicamycin to induce a stress response. And so you'll see that most of the cells that are yellow are grouped together and in one location in this plot. That's a 3D plot that's been flattened. But you will see that down with the black cells called dead in media, there are some yellow ones. Well, this is the heterogeneity in the patient. That amount of tunicamycin causes stress in most of the cells, but it kills some of them. And some of them, which are over behind the cyan colored ones, are much less affected by the drug. So if you add more drug, you get more dead ones, and less drug, you get less affected ones. So you have to balance that to be able to identify the stress response. So on the next slide, we can use this to identify patients and what any one particular patient is doing. And this is without any kind of drug treatment. We just take the cells, put them in media that mimics the bone marrow. And then for any one patient, we can say, in this case, next button, please push. This particular patient has 15% stressed cells, 20% stimulated cells, 10% dead in the media cells. So that's, for those who are interested, that's necrosis. 5 DID cells, that's apoptosis. 2 cells have just been altered, and that's a proteasome alteration that we use, but it's just a strange alteration in the cell. 15% are quiescent, and 25% are the cells that you would expect to see all the time. So each patient has a unique combination of these different cell types in their circulation. So can we use that then to identify cohorts of patients? Next slide. And the answer is yes. So what you see here are those different types of cells starting at the top and going down on the side. You can see the DMSOs on the top, quiescence is next, the altered phenotype, then the stimulated, then the stressed, then the dead in media, so necrosis, and the dead in drug, which is apoptosis. And then across the top it's a standard machine learning clustering algorithm that has assigned each of these patients, each one of the columns is a different patient, has been assigned to a cluster. And it's quite easy to see cluster number 4. You can see that all the high color, the red, is lots. Most of their cells have been designated as dead in media. The one beside it, cluster 3, which is the one that I'm most interested in, and I'll tell you more about, they have mostly quiescent cells and stressed cells. Cluster 1 has mostly unstressed cells and apoptotic cells. Cluster 0 is unstressed cells and stimulated cells. And cluster 2, it's just unstressed cells. They're happy in the environment that we put them in. And so you have a certain number of patients in each of these clusters. And then the question, which has now already come up, is do the patients in each of these clusters respond similarly to different drugs? Next slide. So what we do is we test them with lots and lots of different drugs. So we have, this is a standard plate layout in the lab. It's 1,536 little test wells, and we test 700 different drug combinations in duplicate. Push the buttons and we'll have the different things come up for this. So all the drugs that are approved for or in trial for CLL, next. Duplicates so that we have everything duplicated, next. And then the controls that tell us which is what a stress response looks like for this patient or one of the other phenotypes, next. And then we do dose response curves for up to 1 to 3 drugs and combinations. So in total, it's more than 700 different combinations. Next, for each patient. Next. So that's what the images now look like. If we next look at one more closely, it's what you saw before. So for each patient, we would have about 6,000 images. Next. So 4 images per well and 1,000 well for this particular experiment. So 4,000 wells, 4,000 images like this, each with thousands of cells. Next. Okay, so here's our original groups of patients outlined. The top one is the one where they all died, and down is each of the clusters. And each of the lines that's going this way, coming out of the colored bar, is a different patient. So if we now add a drug, we'll add ruxolitinib. Next. There you can see along the line where the patients go. So it's no surprise that in the cluster where the cells are all dead, that's the red pie chart, they just stay dead. You don't come back to life. So that's not surprising. But remember I told you about the stressed ones. So we're going to look at those, which is the one A3. It's the one up from the bottom one. And that's the brown one. It splits into 2. They start out with about 25% dead and about 25% stressed. And as they go across, you end up with more stress response from the ruxolitinib. On the bottom, cluster 2 are the ones that had just DMSO type cells, just sort of normalish cells. And ruxolitinib doesn't change that. They mostly just stay together and stay normal. Now, if we add a second drug, venetoclax, which is a fantastic drug for CLL, you'll see now all of those ones from cluster 3 that bifurcated have come back together, and now all of the cells are either dead, which is almost all of them, or a small fraction are quiescent. So it says that that particular cluster of patients would respond well to a JAK inhibitor combined with venetoclax. I don't have time to go through the others, but the others also have predicted outcomes with different drug combinations. Unfortunately, except for cluster 2 on the bottom, they don't respond to any of the combinations, any of the drugs. So you don't want to end up in that particular cluster. Next slide. So by using this and having these small cohorts, we can then go into clinical trials, which is what we're trying to finance at the moment, picking out those patients which would fall into cluster 3, the ones that get a stress response from a JAK inhibitor, and then combine that with venetoclax to see if we can deepen the response to venetoclax and maybe push it all the way to cure. So that's the approach. That's what we're doing. And hopefully we'll be able to get this into clinical trials. The biggest holdup is both drugs are not owned by one company. And so getting the companies to agree to these trials has been difficult. And that's the end. Thank you.
Thank you very much, Dr. Andrews. And as a reminder, we are going to hold all questions until the very end after everyone is presented, and then we'll be directing questions accordingly after that. So next, let me introduce Dr. Robert Credell. Dr. Credell has been a faculty member at the University of Toronto, a staff physician in the lymphoma myeloma site group at Princess Margaret Cancer Centre, and a clinician scientist at the Princess Margaret Research Institute since 2007. His research focuses on overcoming treatment resistance in mature lymphoid malignancies through insights from functional genomic screens and integrative genomic profiling of large patient cohorts. He has strong interest in uncovering the molecular heterogeneity of follicular lymphoma and identifying novel biomarkers through genetic and epigenetic profiling of liquid biopsies. Over to you, Dr. Credell.
Thank you, Paul, and thank you so much for the invitation. The LLSC has been a very steadfast support of our research, and I feel very honored by so many of participants joining to listen to what you guys have to say. So this is essentially giving an update on this grant that was funded by the LLSC last year, and we've been working on this over the last 12 months. And it is certainly having a focus in machine learning. We have to say as a disclaimer that I'm personally not a machine learning expert as such. However, we generate lots of big data when we study samples from our lymphoma patients. And these big data just lend themselves really well to machine learning and AI type approaches. And I'm certainly very fortunate to have been able to partner up with both colleagues and also trainees who have great expertise in this field. So next slide, please. Yeah, so I have to introduce two concepts. So one is the one of a liquid biopsy, and the other one is the one of ctDNA. And maybe the underlying question is really whether we can use a blood sample to detect and possibly monitor lymphoma without necessarily needing a tissue biopsy. And for many lymphomas, they present mostly as involving lymph node tissue, oftentimes deep inside the body, can be in the head and neck, in the groin, but oftentimes can be deep inside the chest or the abdomen. So taking multiple or serial samples is not very practical. And oftentimes patients ask me, especially on follow-up, whether the blood work that they had just done actually shows that the lymphoma is in response. And I'm often afraid to say that I can't really tell because you're not measuring what actually could tell us about whether the lymphoma is present or not. So liquid biopsy is essentially a fancy way of saying that we use a blood sample to look for material that is in the bloodstream and that is related to the tumor, and that potentially could offer an alternative to traditional tissue biopsies. And within the blood, we mostly focus on a fraction of the blood, the liquid fraction of the blood, so not the cells, but the actual plasma. And within that plasma, we have lots of proteins and other molecules, but we focus mostly on genetic material and specifically DNA. And that DNA, when it is derived from cancer cells, tends to be abnormal because it tends to have mutations, and it can also have chemical modifications such as methylation modification I'm going to talk about. You can see in this cartoon that tumor cells in green, they have these little round dots, means that these cells, they form apoptosis, they can die off, and they can shed this abnormal ctDNA in high concentration into blood, which makes it possible to actually detect it there. Next slide, please. So what are potential approaches for these liquid biopsies or ctDNA detection? So you can imagine that in many cancer contexts, there can be a use case for early detection, and that is mostly useful for, I would say, solid cancers. So especially when you think about screening for breast cancer, colon cancer, maybe in the future could be simplified with a blood test. Similarly, other cancers that are very difficult to detect early, such as pancreatic cancer, ovarian cancer, could imagine that there's a tremendous hope if there was a simple test that could detect the cancer in a non-invasive fashion. For many blood cancers, this is less relevant, given that many patients with the slow-growing cases actually don't necessarily need treatment up front, and those patients with more aggressive disease have disease that sometimes just probably arises too quickly to be able to detect it early. So the use cases that we think are useful for lymphoma in particular are, for example, the one around rapid diagnosis, because many of our patients still need more than one biopsy to come to conclusive proof that there's lymphoma. So we think that if there was a way of having a simple blood test that can establish lymphoma diagnosis, that could be a useful thing to have. Certainly also there can be a use case for monitoring of treatment response. So more and more we know that as patients go along in their treatment, we can actually track how well the treatment works, and the depth of response we can achieve, even with just 1 or 2 cycles of chemotherapy, can be a very strong predictor of how likely the patient is going to be cured. And possibly we can also use blood samples to detect very specific, unique changes to each patient's tumor, more in a setting of personalized care, so that each patient might benefit from a specific type of treatment down the line. Next slide, please. So this is a cartoon. This is again showing the plasma fraction that is at the top after these tubes are spun down. And in that plasma fraction, we can detect all these molecules, including DNA derived from the tumor. And we'll focus on detecting either mutations or other changes, such as methylation changes. And very important, the tumor-derived changes, they provide kind of a fingerprint, a pattern that we'll try to detect, and that differentiates the sample, the blood sample from a patient with active lymphoma from a patient or another person who is otherwise healthy, does not have lymphoma, any other cancer for that matter. Next slide, please. So in this grant that was funded by the LLSC, actually proposed a method that was developed at our center at Princess Margaret Hospital by Danielle de Carvalho's group, and they had a paper published in 2018 that showed that this method worked very well to detect many different types of cancer, but they hadn't had a chance to look at lymphoma in that paper, and that is a gap that we wanted to address. This technique is called CF-MEDIP-seq. Seq just stands for sequencing, because ultimately we sequence the DNA sequence of these abnormal molecules. CF stands for cell-free, because in the cell-free portion of the DNA in the plasma. And MEDIP stands for immunoprecipitation of methylated DNA. So you can see on the right hand that you have a DNA molecule in black and red, and some of these letters in the genome are actually modified, both in cancer and normal cells, just in different patterns. And one of these modifications is called methylation. That's what this ME stands for. And that specific abnormality is pulled down with an antibody, and so we enrich essentially for that abnormal signal, and that's then gets sequenced on kind of high-performing sequencing machines. And you get this output shown at the bottom right, where you have essentially a read through the whole genome, where certain areas of the genome have very high signal and other areas of the genome have very low signal. Next slide, please. So for this study, we accrued a cohort of close to 250 patients, so 248 samples, 1 sample per patient. Actually, 196 of these were patients. The other individuals were healthy controls. And we performed quality control of the data, after which we had 215 available for analysis. And very importantly, we included different types of lymphoma, and those are mostly lymphomas that are present really within the deep tissue, not so much in the blood, both across non-Hodgkin lymphoma and Hodgkin lymphoma. Within non-Hodgkin lymphoma included diffuse large B-cell lymphoma and follicular lymphoma. So these 3 lymphoma types together represent about 50 to 60% of all cases with lymphoma, so the majority of patients. And importantly, when you do these studies, you need a validation cohort because you can build very fancy statistical models in the discovery cohort, but you need to be able to validate it on an independent data set. Next slide, please. So what do these data look like? This is just a very simplified illustration. So we have different genomic windows that's in rows. So you have different chromosomes, different coordinates. Each of these regions is essentially 300 letters long. And within those regions, we measure the signal of methylation. And you can see in the very first row, that's a region with high methylation, and that is predicted to be high in cancer. In the second one, it seems to be low. It's predicted to be similar in both cancer and healthy patterns. And then at the very bottom, we have a region that is highly methylated in cancer that is only detected in cancer samples. Of course, when you see this, and I will ask you which of these regions is important to detect whether someone has cancer, this is not you tell me easily, you pick the last one because it's very high in cancer. And if you only had 4 regions, that would, of course, be very easy to do. Next slide. In reality, we don't have 4 regions. In reality, we have millions of regions. And we, of course, narrow this down. We filter some of the non-informative regions out, but we still end up with hundreds of thousands, or tens of thousands of these windows. And as humans, we just couldn't comprehend that much data. So we need essentially machines, computers to help us identify complex patterns. So we need essentially to apply machine learning. We need to apply statistical models. And statistical model is just kind of a smart equation that allows us to make sense of the data that is otherwise really noisy or messy. And you can think, for example, of, you probably checked your emails today, and as myself, you probably get annoyed sometimes that you get junk email, but there are actually filters behind the scenes that filter much of the junk email out. And so those filters, they are built on statistical models that identify patterns of what a junk email looks like compared to a true email that you should receive. And similarly here, we try to essentially identify these patterns, and only the computer can do this. We could not do this in our mind or just by hand. Next slide, please. And there's many different models that one can apply. More recently, many of the cutting edge models, such as neural networks, are very popular. And those are, for example, the models that allow you to, if you look at your phone and you go to the photo app, you can click on a button and get all the pictures, all the pictures that show your cat or your dog if you have a dog. And so there's very complex models in the background that identify with quite high precision whether an image is derived from a cat or from a dog. But oftentimes these models, you can't really tell exactly what specific features in the picture actually leads to being classified as one or the other. And we applied this, actually, not one of these more recent models. We applied a very old school model called generalized linear model that takes as an input the features from methylation that I showed you in the last slide, but then gives us the prediction in the validation cohort of whether this case may belong to either a lymphoma patient or to a healthy control. And the good aspect of this very simplified model is that it tells us specifically which features are important. So if you wanted to understand in the future better why certain features, why certain methylation segments in the genome are important, we can potentially study these in the lab and learn more about the biology of lymphoma. Next slide, please. And I'm going to show you over the next few slides what an output looks like. And this output looks fairly dry. It's a table with numbers. And this is a comparison of lymphoma versus control. And this gives us different sort of metrics that are all derived from our validation cohort. So essentially, all these metrics tell us how well the model works or how well maybe it doesn't work. Next slide, please. So then the first reaction is, so what? What does that all mean? Go to the next slide. Yes. So, and out of the many metrics, probably the one that is easiest to appreciate is the one that we call accuracy. And this just essentially calculates out of, let's say, 10 cases, how many were accurately classified into either lymphoma or control cases. And in this case, this model actually has an accuracy of about 0.9, which means that 9 out of 10 cases are classified correctly. So if you were to apply this test in the clinic, 9 times out of 10, it would make the correct diagnosis. Next slide. But other metrics are important as well. So there are 2 pairs of metrics, sensitivity and specificity. So first, sensitivity. This shows us that the model is quite good at catching lymphoma. So we miss very few cases. So the sensitivity is about 96 to 99%. So it detects almost all cases with lymphoma, even those cases with early stage disease. Next slide. And that comes at some trade-off, because the higher the sensitivity, the lower the specificity will be. So you can see that the specificity is lower, especially to detect lymphoma. The specificity is only about 68%, which means that this model, the way it is designed as of now, would be very good at catching all lymphoma cases, but at the expense of misclassifying some healthy controls as having lymphoma. So then it essentially means that this model on its own requires an additional follow-up test to confirm with certainty whether a person has lymphoma or not, and typically that would be a tissue biopsy. Next slide, please. So you also have the positive predictive value. So if the model says that someone has lymphoma, it is usually right, and this positive predictive value is in the range of 80 to 90%. You also have the negative predictive value. Next slide. Which is in the range of 90 to 98%. So if the model says that someone is healthy, it is almost always right and will rarely make a mistake. Next slide, and getting to the end. So I just want to give you kind of one example of what we can learn from applying these techniques to plasma samples from lymphoma patients and from controls. There's many additional steps that we're currently pursuing. First of all, we would like to add additional lymphoma subtypes. I showed you 3 lymphoma subtypes, but there's up to 80 different lymphoma subtypes, some of which are very uncommon, but others are more common. So that's one of the goals, to see how well the models may perform for more rare lymphoma subtypes. We're still working on potentially refining the model and finding different ways of either doing the actual assay or different, maybe more innovative machine learning techniques that we're currently exploring. We'd like to also interpret the biology. So this data set gives us a lot of insight that certain part of the genome are important, likely for biology, but at this point, we do not know exactly why that is. So I think that if we were to understand these things better, we might understand more about how lymphomas arise in the first place. Importantly, we'd also like to apply these models to samples after treatment to detect how well treatment has worked, and specifically to apply it to a setting or to detect minimal residual disease, which would apply to patients who have had a good response on CT scan, but may not have achieved a complete response, which we could only detect by these refined techniques. And ultimately, of course, would like to advance these tests into the clinic, but certainly a bit more research to be done before that is the case. Next slide, which is just my acknowledgement slides. So I'd just like to acknowledge that this work, of course, could not be possible without the very generous support of our funding agency, most notably the LLSC, and certainly also the patients who generously donate their blood samples for this research. And as you can imagine, behind the scenes, there's a whole team of people involved in making this research possible, both academic colleagues at our institute, but also many trainees who work on these data as part of their studies. Thank you so much for your attention.
Thank you very much, Dr. Crudell. Next, we'd like to turn our attention to a pair of researchers, Dr. Sabine Mai and Dr. Yuval Gurini. Dr. Mai did her master's and PhD in Germany at the University of Cologne and Karlsruhe, respectively. I don't think I pronounced that correctly, but her postdoctoral training in Switzerland, the Basel Institute for Immunology, and took an assistant professor position at the University of Manitoba. She set up her lab at the University of Manitoba with a focus on genomic instability, cancer, nuclear architecture, and translational research. Professor Yuval Gurini heads the Nanodynamics Lab at the Faculty of Biomedical Engineering. Trained as a physicist, his research spans a broad range of biophysical topics, both fundamental and applied, including genome organization with the nucleus, DNA origami-based nanotechnology, and the integration of spectral imaging and artificial intelligence for genetic and pathological applications. Dr. Mai and Dr. Gurini will talk about her funded research. She's automated AI-assisted spectral detection of single multiple myeloma cells. Over to you.
Thank you, Paul, for the introduction. As you said, this project is done in collaboration between my lab here at the University of Manitoba and Dr. Garini's lab at the Technion. And two of our students were involved, Dia Dutia here in my lab and Yotam Shapira in Yuval's lab. Next slide, please. Our focus is on multiple myeloma and the challenges and unmet clinical needs of this disease. Unfortunately, multiple myeloma remains an incurable blood cancer with a 5-year survival of only 50%. The incidence is in Canada, and these are the numbers I'm talking about. 11 Canadians are diagnosed with multiple myeloma every day, with a prevalence rate similar for men and women. And in the last 30 years, 1 in 2,505 Canadians was a myeloma patient, and that hasn't changed. The Canadian Cancer Society estimates 4,100 Canadians diagnosed with multiple myeloma in 2024. What is the most urgent need? We need better detection and monitoring of the patients to be able to develop a cure. How can we achieve this? The focus has to be on early disease prior to the full-blown myeloma. It has to be on risk stratification and on disease monitoring early, as well as after treatment, including during the stage of minimal residual disease, MRD. Next slide. So our project outline was, we reasoned that spectral imaging and computer-assisted AI can aid in single and multiple myeloma cell detection. And our vision in this project is to revolutionize myeloma patient care. Objective 1 was to train the computer AI in the recognition of normal and blood cells versus myeloma cells. This had never been done, and we needed to develop a training concept. 2, we needed to determine the detection limit of myeloma cell identification after training in Objective 1. And finally, take what we have learned, or what the computer and AI have learned, onto patient samples and see how good we are in detecting the multiple myeloma cells in real life. Next slide. So the methods, we performed cell detection via modified Gimsa staining and immunophenotyping with antibodies specific to multiple myeloma cells. And for the cell detection, we worked on automated scanning and computer-assisted AI. Next slide, please. So how did we train the system? We started with 100% myeloma cells and 100% normal mononucleated blood cells, primary cells that were stained with modified Gimsa. The myeloma cells we used for the training were patient-derived and a cell line. They were so-called pure cells because 100% of the cells in culture were then myeloma cells. And first of all, the system was trained in learning how myeloma cells look like as opposed to normal cells. Next slide. After that, we trained the system to identify myeloma cells in spiking experiments, where we began with 1:1, 1:2, 1:10 ratios. Here we show 1:10 and 1 in 1,000. And you see both the Gimsa stained and the CD138 stained, which is sufficient because this was a myeloma cell line. We just confirmed by CD138 that the cells detected were indeed myeloma cells. Next. We continue to train at 1 in 10,000, 1 in 100,000. Next. Next slide. And finally, we asked whether the system was able, after training, to recognize the myeloma cells in a multiple myeloma patient sample. And what you see here on the left is modified Gimsa again on a bone marrow sample from a myeloma patient. The majority of cells, blue cells in the image, are myeloma cells. One in the center is binucleated. then you see a lot of red blood cells and you see small lymphocytes as well. On the right-hand side, you see the CD138 fluorescent immunostaining. Next slide. Those slides were sent to Yuval's lab, and Yuval will continue now to explain what were the next steps in the recognition and training of the AI.
Okay, thank you very much, Sabina. Thank you everyone for taking part in this important webinar. So we are getting the slides and we are scanning them. I should mention that we have two different ways of scanning. One is more traditional with a kind of commercial system that scan the slide exactly in the same way that we can see it by eye. We sometimes call it a red, green, blue image, just gives a color image. The second way of measuring the slide is with a very unique system that was developed in our lab with a method which is called spectral imaging, that very briefly it measures for every point of the image an information that is almost invisible to the human eye. We cannot see it because it has a very fine ability to detect very small changes in the color. And by doing such a measurement, we can actually get more information than can even be viewed by a pathologist or an expert that is looking at the slide by eye. So after...
He's muted. You're muted.
Sorry, next slide, please. Sorry. Yeah, so after the measurement, this is just an example of a typical spot that, as you can see, has a diameter of approximately 5 millimeters. Of course, it's a very low magnification, so here we do not see details, but the image is made from many, many small images that we can identify a lot of the cells. Next slide, please. So this is a zoom in for one area of the image. And here you can definitely see that we can image cells quite nicely. And more particularly, we are interested in the nuclei of the cells. A lot of the analysis that we are doing is based on the nuclei, as we figured out and we found that the nuclei, the size, the shape, whether there is a single one or more than one nucleus per cell, gives a lot of information for identifying the tumor cells. Next slide, please. So then we go to a process that is very well known in imaging, which is called segmentation. Each one, as a human being, can very easily do that. It just means to take a pencil and to draw the line around each one of the cells. So for us, it's very simple. Our brain is excellent in doing this operation, and even a young child, after some practice, could do that quite nicely. For training the computer to do that, that's a very hard task. And of course, that we are talking about something that must be done almost perfectly to, in some cases, millions of cells for each cell. Next slide, please. So what you see here is, again, a zoom-in picture of the result of the segmentation. So the black and white image, it just shows you where are the cells or the nuclei that were identified by the system. And on the image to the left of it, what you see is exactly the same image, but this time, instead of showing the white spots, we really show how the original image look at that spot. And if we compare the right image to the left image, we can see that the computer did a pretty good job, and it identified almost all the nuclei that exist in the image. Again, it's never really perfect, and there are all kinds of problems. Sometimes there are very small points that were identified that most likely are a kind of a debris or something which is irrelevant. It's not really a cell, maybe a red blood cell, but it's not the most important thing. Next slide, please. Okay, so again, we show the same image. You can see, just for a comparison, the original image and the way that it was below that. It shows the way that it was segmented, as we call it. It shows what the computer found as the nuclei in the image. Next slide, please. And okay, so the first most simple kind of analysis that we do is to measure just the size of the nuclei. Now it's clear to everyone that just taking the size of the nuclei and the cells by itself is not a good enough parameter for identifying myeloma cells, but what we see here, what we found here is that it's an excellent starting point for identifying quite nicely many of the myeloma cells. Now, just briefly, what we see here in the red bars, it shows the size of those cells that may be large enough, so some of them may be myeloma cells. And what we see in the blue bar, you can see that this is lying on the small numbers, which means small size. So this shows the populations of cells which are quite small. Next slide, please. Now, just by even looking at those two different size distributions, we can see that if we put a kind of a threshold, if we say everything which is approximately above 250 pixels, points from the image, is big enough, and we want to identify, first of all, what are those cells versus those cells that are smaller, that are lying left from that line. Next slide, please. And when we kind of filter out, just by using this operation, you can see what we are left with. So what you can see is that most of those small points, whether those are, again, red blood cells or debris, actually disappeared, and we are left only with the bigger cells, hopefully the myeloma cells. Next slide, please. Okay, Yotam, maybe you want to say a word about it. What we see here, I'll start, is the statistics of what we get, and let me explain what we show. So on the left side, we see the different types of samples that we measured, and the second from the left column show the total number of cells that were identified on that slide by this operation that I called before segmentation. You can see that we are dealing with tens of thousands of cells. The next column shows how many cells were identified as myeloma cells out of the cells, all the cells that were identified. And on the right column, it just gives us the percentage of the myeloma cells that were identified out of what we know is the real value. So, for example, the first sample, the first line at the top, that slide contained only myeloma cells, which means 100% of the cells were myeloma. The system identified only 84 point something cells. It's not perfect, but it's not bad. The second slide, as you can see on the left, contained only normal cells, and the system identified only 17 cells as if they are myeloma cells. Of course, it's a mistake. There are no myeloma cells on this slide. If we translate it to percentages, we see that this is a very small, very low percentage. And then we go on, you can see a sample that contained a ratio of 10 cancer cells to 90 normal cells, 25 to 75, etc. And again, the numbers that were, the percentages that were identified is not bad. Next slide, please. Okay, so now what do we do next? We already see that the system is not doing a perfect job. So the next thing is, in a kind of an application that is aimed for future use by pathologists, the user can very easily, after doing the analysis, ask the computer to show on the screen all those cells, or some of them, that were identified as myeloma cells. So the first case is not very interesting because we know that most of the cells are myeloma. The next case is interesting because, as I showed before, here all the cells are normal, but the system identified 17 myeloma cells wrongly. Okay, so now what the application can do in a very simple way, just by having the expert clicking on a button, is to show typical images which are zoomed in of the cells that were identified. So, for example, if one would look like that, to say, yeah, of course, these are myeloma cells. But for example, if someone would look at that, we'll say, well, this is wrong. This is not even exactly a cell. It's a kind of a mixture of a kind of an aggregate of cells or maybe debris of the stain. So he will just say, okay, this is nothing. We'll remove it from the list. And then it can go on. You can see that there are cases like we see at the bottom right, where the system wrongly identified two neighbor cells as if they are one. So here too, if the expert would look at it and say, no, this is not a myeloma cell, in a relatively simple kind of control step, the expert could actually check the system, test it, and get to the exact conclusion according to what he or she finds from that. Yotam, I'll let you continue with the most interesting cases at the bottom.
Yeah, thank you very much. The slides, the image is supposed to be a little lower. The three images that we see on the bottom are from the 1 to the 100,000 slide. It means that we have a very, very low concentration of myeloma cells. And using the system, using the AI, we were able to detect the only two myeloma cells on the slide, just using the size threshold that Yuval talked about earlier. And there was one misclassification of these two tightly packed normal cells. So we are able very easily, it's a model that was trained on a very small sample size of nuclei, and it can be tremendously improved in the future, very easily actually, to detect very, very small concentrations of myeloma cells. And then the point is that the physician or the pathologist can automatically scan and train the model and test the model over lots of samples, and the system will automatically give it the suspicious cells, those that the system determines as myeloma or cancer cells, and it will only have to look at it and say, oh, we are correct. We found the myeloma cell that we were looking for in a very small concentration, instead of looking at all of the slide and looking really nitpicking the correct place. So this could expedite the entire work in a tremendous way. Thank you. Yuval, do you want to continue?
Okay, next slide, please. Okay, so here again you can see one of the typical cases, and you can see that, of course, that when I talked about segmentation and using the threshold, all these procedures are based on artificial intelligence algorithm that we developed specifically for this case. And you can see the identification of those target cells in real patient samples. And the red arrow shows a typical red blood cell. What we see on the left is just a typical image from the slide itself. And after some processing, the images from this spectral information that I mentioned before, we can get images that look what we see on the right side, which are easier for the artificial intelligence algorithm to identify the myeloma cells, together with verification, as we see at the lower image. Next slide, please. Sabina, I'll return things to you. Thank you.
Thank you, Yuval and Yotam. So in conclusion, this short summary shows you that we were able to train the system to recognize myeloma cells. Next. Myeloma cells were identified by computer-assisted AI. Next. Myeloma cells were detected also in patient samples, not only in spiking experiments. Next. And it is necessary that a hematopathologist reviews the cell selection made by AI and that they work together to enhance a diagnosis. Next. Future work will continue on patient cohorts and in collaboration with hematopathologists. Next. The patient impact. This new method of cell detection will assist in early detection, in the assessment of minimal residual disease, and in disease monitoring. It will ultimately be based on liquid biopsies and allow for easy monitoring while avoiding bone marrow aspirates. It is expected to enhance myeloma patient survival and enabling a cure. Next slide. Thank you very much. Without the funding from Leukemia and Lymphoma Society of Canada, none of this would have been possible. Thanks so much.
Thank you, Dr. Mai and Dr. Carini. And so let me introduce our last but not least panelist, Dr. Michael Raw. Dr. Raw is a clinician scientist at Queen's University and Kingston Health Science Centre. His combined MD PhD training was conducted at the University of British Columbia and Terry Fox Lab of the BC Cancer Agency. His specialized hematological pathology training was conducted at the University of Toronto. His clinical practice and research focus is centered on myeloid blood cancers, particularly understanding their genetic and immune origins. Over to you, Dr. Raw.
Thank you, Paul. Thank you to the LLSC, this opportunity and funding, and everyone attending today. And it's been really interesting to hear about my colleagues' work as well. So I'm going to talk about a preclinical precancerous condition called clonal hematopoiesis. Next slide, please. And so DNA sequencing technology, which we heard a bit about from Dr. Kreidel, is really enabling us to find the drivers of cancers, and even in the blood in people that are asymptomatic. And I'm going to talk about this condition briefly. It's called clonal hematopoiesis, or CH, or CHIP for short. In addition to the fact that it increases the risk of cancer, it's the very start of cancer, we're also interested in it from a health consequence because it contributes to inflammation and diseases of aging that are associated with inflammation. And so what we're hoping to do is to access and apply AI to routinely available blood cell images in the lab. We were just hearing about some of those images now, really exciting things, to help screen for this precancerous condition. Next, please. So I'm a blood pathologist or hematopathologist. We were hearing about those in the last talk as well. Our research and clinical aspects are focused on the myeloid neoplasms, which include myeloproliferative and myelodysplastic neoplasms and acute myeloid leukemia. As you'll see next, our job starts when the patient comes to the hospital or is referred to the hospital. And at this point, they have abnormal blood counts. They have symptoms maybe related to the abnormal blood counts. And we are looking at peripheral blood, but quite often we're needing an invasive sample from the bone marrow to apply a battery of tests to diagnose these. And more now, next please, molecular and genetic techniques are coming into play. Next slide. So this is an example of a driver gene in acute myeloid leukemia. It's called DNA methyltransferase 3A, or DNMT3A. This is a schematic of the protein and its structure. And on the top, we have these lollipops, and they represent genetic hotspots for mutation in AML. So for example, on the right, the green one shows us arginine 882 is a frequent spot of mutation in AML patients in about half of cases. Next, please. We're applying these genetic sequencing in these patients that are coming into our hospital walls and finding, for example, an AML patient with this DNMT3A hotspot who also had another mutation. And we're doing our very best, guiding the clinicians and trying to treat them. This patient had an unrelated or allogeneic bone marrow stem cell transplant, but is subsequently suffering from chronic graft versus host disease. One other example next is a 55-year-old woman who had a different arginine mutated, just a little further down the protein, arginine 736, but also 3 additional mutations, so more complex genetic architecture. It required multiple therapies for our clinicians to get that patient into remission. And unfortunately, there was a relapse after her allogeneic bone marrow stem cell transplant. So I illustrate these because I think we can do better. we're engaging with patients when they have genetically complex disease. But I was talking about this early clonal hematopoiesis. Next slide, please. These exact same two DNMT3A mutations in AML cancer patients can be detected in healthy people in the community, in this case, in Toronto, in the study that we conducted of anonymized adults 65 years and older. 9% of them had detectable mutations in the same cancer driver gene, DNMT3A, although they showed absolutely no changes in blood counts and no symptoms. So it's a very cryptic, silent state, and this is the clonal hematopoiesis that I told you about. And why this is that over time, our hematopoietic stem cells accrue mutations, they accrue genetic damage with aging. Sometimes these land in an oncogene like DNMT3A, and these are the very origins of cancer, and this is what CHIP or CH is all about. Next slide, please. This is surprisingly common. Next, please. And in our case, we found it in 23% of adults in 65 years and up. It's dominated by mutations in DNMT3A, which we just talked about, and another regulator of epigenetics, or the methylation of DNA, called TET2. Next, please. You can see that in this histogram here, where TET2 and DNMT are by far the most common drivers of this cryptic clonal hematopoiesis with aging. On average, the mutant clones represent about 20% of blood cells, and so there's a mixture with 80% normal cells. But there's enough of them there that we could detect it with a simple genetic sequencing test of blood. Next, please. Just as a schematic to show you more about CHIP. So on the left, we see some gray dots. These are normal stem cells. They're accruing some damage with aging. So for example, in the bottom, there's a green dot that appears in one of the hematopoietic stem cells, a blue in another. They're not causing any issues. But in the middle of the panel where we see the red, which is a TET or DNMT, that now allows that stem cell to get an advantage and to expand and to outcompete the other stem cells. Next, please. And maybe one more click as well. So what happens, though, is to progress to full-blown cancer, you need additional events. So to go to MDS or AML, as shown in that schematic, you need other hits. But that first hit, that CHIP hit, is what sets the ball rolling. And overall, in a lifetime, these people have a risk of cancer of about 11-fold increase compared to normal, or each year that they're alive, about 0.5% or 1% chance of progressing each year. But despite that, the majority of people with CHIP are not going to progress to cancer. There's a minority of them, and we want to understand better about that. Next, please. So the other reason we're interested in CHIP, as I mentioned, is that the mutations cause inflammation and inflammatory diseases. Next, please. So in red, if we follow, there's a stem cell in the marrow that's red and a mutant. It's carried by its mature progeny, like macrophages and neutrophils. These are immune cells. They're going to increase inflammation in the body, and in a vicious cycle, contribute to inflammatory diseases and further clonal expansion. And this is why CH is also a new risk factor for other diseases like heart disease, lung disease, kidney disease, infectious disease, and we're starting to study as well solid non-hematological cancers and their occurrence and their progression. So a lot of clinical interest in CHIP and its health consequences. Next, please. Some other opportunities that we're facing around the time of this grant application was there's a lot of people with CHIP, but we're getting better at predicting who among those people are at greatest risk of progressing to full-blown cancer using scoring systems that factor in age and the clone size and the type of driver. Now, can we do anything about it? Well, it's looking hopeful. There are some low toxicity treatment strategies that are emerging that might target these CHIP clones at the very origins. And so all of this is shifting our excitement and the idea, can we, like Paul was saying at the start, shift the needle? Instead of waiting till people come to hospital, can we find blood cancers early? Can we do blood cancer prevention and health optimization? That's exciting, but next, there are some challenges. Of course, it's not ethical, it's not practical or economical now to go sequencing everyone and screening for CHIP. And usually, as I mentioned, people with CHIP don't have any symptoms, and their blood counts overall are relatively normal. Next, please. So what I'm going to finish up on is to tell you briefly about how we're going to be applying AI to digital blood cell images to help us maybe screen for CHIP. Next. This is a screenshot of a program that we have in our lab where when the blood cells run through the analyzer, the counts are performed, and individual white blood cells in this case are being captured. So at the top, we have some neutrophils here, some lymphocytes and monocytes at the bottom. Next. We know in MDS, so further along from CHIP, that there can be changes in neutrophils that are recognizable by the technologists and the pathologists, so they get less lobation of their nucleus. They have less granules that we see in their cytoplasm. And so it's not a stretch to think we might be missing some subtle changes in clonal hematopoiesis because a lot of the drivers of MDS are the same as the ones we see in CHIP. Next, please. Looking at red blood cells, for example, next. We know that in MDS, they can be larger, they can become more oval shaped. So again, are there some changes we maybe have not been recognizing or beyond the capability of humans to see, and a machine could see much better that might help us to recognize CHIP earlier? Next, please. So I'm not an AI expert, partnering with those in this grant who are, including a dynamic med student now at McMaster, Akshaj Darbar, who came out of our biomedical computing at Queen's and was working with us. Two professors here, Dr. Catherine Terishkin and Ting Hu, who are affiliated with our computing department and pathology department. They have expertise in this area, and especially around applying machine learning to bioinformatics and to opening up the box, if you will, and understanding AI algorithms and having them be interpretable, which I think is important for us to understand with CHIP. Next, please. So briefly, what we've been doing to this point is accruing all of our samples, and with the help of research associate Dr. Amy McNaughton in the lab, sequencing these with a targeted panel for CHIP. So we'll know if they have the ground truth, if they have CHIP or they don't. We're including some people with full-blown myeloid leukemias to have that side of the spectrum for training the AI, and also some other conditions that are commonly known to affect blood cell counts and morphology, like iron deficiency, kidney disease, for example. So we can bring all of these into account and look for CHIP-specific effects. Next, please. And so far we have sequenced 360. We have access to all the samples we want. We're seeing the expected proportion of CHIP. In this case, overall, it's around 25% of the subjects who anonymously were getting their leftover blood from routine testing in the lab. So we're well set up to be looking at these. We have their digital images. Next, please. There's a three-pronged approach to the AI. The first is that we're going to extract in panel A at the top some features from the images, some metrics, and look at, as you've been seeing, some of the mapping of these now. In the middle, we're going to look at more individual cell images. And on the bottom, next please, which we think will be the best approach, is a multiple instance learning approach, where all of the cells in the image are going to be grouped into a bag, and we're going to be classifying the whole bag and training based on this whole of cells. And this mimics more like what a technologist or pathologist is going to do, assessing individual cells, but putting it all together to come to diagnosis. Next, please. So a little bit of progress. We're taking them out of the clinical system, so we have to be able to now re-identify some of these cells. Akshaz has applied the algorithm, the ResNet, to a published set of images. And so with training here and testing and training, we have a fairly accurate way of identifying these now. Next. And then you heard about some of the features that are going to be steps required in the last talk, so I won't spend time on this too much, but we're going to differentiate foreground from background, remove noise, highlight the nucleus, highlight the contours of the cells, as you were hearing about. Next. And so then what we really want are features like on this slide here, where humans are going to be looking for some of these, but there's many, many more features and subtleties that probably the human eye is not picking up on, and especially with the thousands of cells available. And so these are features that are going to be extracted. Next, please. And so to wrap up what we're going to do next, and we're excited to be underway with this summer, is doing the feature-based machine learning, looking at the individual cells, and then also putting this all together, mimicking the human sort of approach with multiple instance learning. Next. And so hoping to push the needle, I've showed you some examples of patients above the line who have leukemia. We're really doing our best there with the current clinical focus. But on the bottom, next, we know there's this cryptid period where people are having normal cells and gradual accumulation of mutations, gradual phase that takes many years to get to full-blown leukemia. And we're hoping that AI can help us to tease apart and identify those people for blood cancer surveillance and healthier aging. This is going to require a lot of thought moving forward, patient consent, and pushing the needle towards primary care and guiding family practitioners, and maybe screening patients in the future for specialist care. Next. So I really would like to thank again the Leukemia and Lymphoma Society of Canada and the UFCW as well for funding support, members of the lab and our collaborators here who I've mentioned. Thank you very much.
Thank you very much, everyone. If you're not on camera, can you please, as all our panelists, put your camera on for the Q&A period. As everyone can see that's been listening, this is an extremely detailed information, just doing detailed presentations. A lot of great information here. So if you do have questions after the fact that you've thought about it, after you've had time to absorb the information, please let us know. In the interest of time, what we're going to do, I think we'll do one question each, and then we'll wrap it up because we don't want to keep everyone waiting too long. So I think what we'll do to start, we're going to start with Dr. Ra, as he was the last speaker. And I think you touched upon this, but can the models that you're using distinguish between a benign chip versus chip that has a higher risk of progressing to a blood cancer?
So that's an excellent question. Our initial approach is going to be just more general, asking if it can identify chip or not. But obviously we are interested as well in breaking it down by driver gene, or some of those higher risk features. That would be the ultimate goal, is to be able to tease apart not only chip but the higher risk chip. What we do also have is the ability to apply sequencing tests that would give us more of that information. And so if this model is able to predict chip, that's a really helpful start to begin with, to narrow down who might get that more refined genetic testing. But we're really excited to see, Paul, if we are going to be finding features of higher risk chip. So we are tracking that information as well. I didn't have time to sort of mention that in the talk.
And do you think this will be used for predicting adverse outcomes as well that are associated with chip, or for lifestyle or environmental factors that may be?
Yeah, potentially. So in part of the grant, what we know is that in the community, like a lot of the work is done by private labs, in the community, and they use similar imaging technology. So we're hoping that we could roll this out, with some validation here and partner with primary care. What ideally we would have is some guidance to help those family docs, who are dealing with so much and are overworked and under-resourced. Evidence-based kind of pathways to guide them to referral. Like when should you refer someone? We've got an unexplained anemia here. The MCV is climbing. To really try to help those clinicians and patients who are in primary care to help standardize and make more equitable access to kind of hematology and these sort of earlier diagnosis and interventions.
Great, thank you. I mean, there's obviously a lot of questions around how this is going to be integrated across the board, and I think that's something you're all thinking about as you're developing these technologies, is how this is going to be integrated into the everyday supports that are available for people that are affected by blood cancers. Thank you very much for that, Dr. Ra. And so to Dr. Mai and Gurini, you spoke about detecting multiple myeloma as a whole, but do we think we'll ultimately be able to distinguish between different subtypes, or whether we're looking at something like high-risk smoldering myeloma that may progress to active disease, or along those lines? What were your thoughts on that type of detection?
Using AI, you're asking?
Yes,
Or in general?
yes.
Okay, using AI. Together with the Gurini lab, we have done work not in myeloma about the risk prediction or stratification of patients, but in a different cancer, in a solid cancer, using also the spectral scanning approach, and that's been thyroid cancer. There we were able to differentiate between the individual histotypes, and some of them are more aggressive than others. So I think the spectral parameters that AI can recognize have the potential to identify the risk of a patient to progress, like from smoldering to full-blown myeloma. Yuval, what do you think?
Yes, so I think the answer is yes. But the more delicate question is on what price? Because let me distinguish here between measuring in bright field a stain like H&E or similar, which is very simple, it's very fast, it's very cheap, and but in a way it's most likely also a bit limited in the ability to detect subtypes. Now, I think if we want to detect them after initial detection of cancer, then beyond any doubt, the best way to go still in imaging is to use antibodies, whether in immunostaining or in fluorescence. And we are already doing that, using even this spectral imaging. We enter to this field of maybe have the title spatial biology, where we can label up to 10 different antibodies on the same sample in one drop, more or less, a very simple procedure, but it is fluorescence. It is more expensive, it takes much longer time to measure, and I believe that you would have to tell the experts would have to tell which are the right antibodies for identifying which subtype. I believe that by measuring and identifying those, subtypes can be detected in quite a nice manner.
Thank you. I think this speaks to the development of the technology and improving the specificity and everything of all our detection and imaging technology that we have, so that is applicable across the board in every institution. Dr. Cradell, so you spoke about using cell-free DNA to help to detect lymphomas. Why is that approach used versus regular blood samples? What are the advantages of using cell-free DNA versus just your typical blood slides, I suppose, or blood samples?
Yeah, I think the key is that the lymphoma cells, as any other cancer cell, would shed DNA into the blood circulation. So you kind of really have to go after the cell-free DNA fraction. Typically, in most patients with the most common forms of lymphoma, there's not really a high proportion and sometimes no circulating abnormal lymphoma cells in the blood at all. So this is very different from, let's say, patients with leukemia in whom the disease most is characterized by abnormal circulating blood cells. So those you can catch, of course, very easily. But that's quite different from patients with most cases of lymphoma or patients with solid tumors. So you really need the cell-free DNA fraction.
Right. Yeah. So it speaks to the fact that the difficulties in the diagnosis across the board, and that, as you said, diagnosing lymphoma from a blood slide is much more difficult than it is from other subtypes or other types of blood cancers. So do you think this will be able to be used for identifying high-risk patients at an earlier stage, than they currently are?
I think the way I see it is that the most pressing applications will be in monitoring of treatment, either during treatment or after treatment. I see already now there's quite a number of clinical trials that are coming up where similar techniques are being applied. So I think we just have to see kind of which specific test will kind of be the winning strategy or will be the most popular one. But yeah, certainly assessing whether the patient has achieved a complete response or not remains very important. And those tools likely can give us the answer in a much better way than any other test before.
Great, thank you. And so finally, we'll turn to Dr. Andrews, and this is a very general question, I guess, but what are the implications of AI-guided treatment decisions over long-term toxicity and quality of life for CLL patients in particular, I suppose?
Yeah, for CLL and probably many of the different diseases, one of the things that you can achieve is to have the minimum effective dose. And so then for any particular patient, you can minimize the side effects by having A, the most effective combination of drugs, and B, knowing what the most effective concentration that you need to achieve is to get the desired outcome. So I think really, and this has worked out to be completely true in a clinical trial that we've just concluded in ovarian cancer, where the patients had a much higher quality of life on treatment than they did with the standard of care.
Thank you. And I think that really lends to the question of what is the long-term impact of AI on the development and design of clinical trials for obviously we're talking about blood cancers here, but I don't know if you have any quick words of wisdom about the the implications on clinical trial development.
Yeah, well, you have to show single agent efficacy to be able to get a drug approved. And if you can use AI to monitor your enrollment into the clinical trial so that the patients you're enrolling in the trial are the most likely subset of patients to respond to that drug, then the odds on you being able to get a good outcome and one that will facilitate approval increase dramatically.
Wonderful. So We'll leave the questions at that for now, and then we do have a couple more slides just to end it. But I wanted to say thank you all for taking part in this webinar today. This is a very complex topic, of course, and I think there's a comment that just came through that I'd like to share. This is the future. Thanks to all for devoting your research lives to this endeavor. AI needs human ingenuity to explore this new horizon in disease management. Better for individual patients and better for cost analysis, because eventual treatments that will never work do not have to be administered, and ones that may work can be given. So I think it's a really nice comment that came in through the chat today. So, Pascal, we have a couple more slides left, I believe, just to wrap up. So we invite you to our next online event, which is next Monday, Advancing Immunotherapy for Blood Cancers with Dr. Natasha Kekri. It's June 2nd at 2 p.m. Eastern. And the next slide, please. Yes. And if you're living in the Calgary region, you might be interested in attending our in-person event on June 5th at the atrium at the Delta Calgary South. Visit bloodcancers.ca or scan the QR code on the screen to have more information or to register, but don't wait because the registration closes on the 29th of May. Please remember that we are here for you. Contact us. Please note that a short survey will be sent to all participants after this presentation. We thank you for taking a moment to respond, as it helps us plan for future events. And finally, thank you all for being with us here today, including the speakers and all the guests, and take care.
Thank you. Bye.